Shall we start, sir?
Hemant, I think we should start the meeting.
Okay, sir. Fine.
Okay,
Shubha Madhyana,
we can
all of
start.
you. Shubha Madhyana, all of you. Myself, Dr. Hemant Pansade, organizing secretary for this Naitaniki webinar September series. We started a particular webinar series on pathology to enhance and update the knowledge of Ayurvedic Vikruti Vigyan PGs as per time. Today we are having eminent personalities for this webinar series. Dr. Mayur Bhosle sir as a guest speaker, Dr. Dipali Amle madam as a chairperson, lovable RVPGA president, Dr. Subhash Wage. Before I start, I would like to invite our spokesperson, Dr. Mrunal Bhoyar madam, to introduce our organization.
Good afternoon, sir. Am I audible?
Yes, yes.
Yes, ma'am.
Okay. So good afternoon and namaste everyone. Myself, Dr. Mrunal Bhoyar. I'm working as an associate professor and HOD of Rog Nidan Department, Parul Institute of Ayurveda, Gujarat. And I am here to give a brief introduction about our association. So our organization, RVPGA, that is Rog Nidan and Vikruti Vigyan PG Association, was formed in the year of 2016 under the able and dynamic leadership of respected Dr. Subhash Wage sir, with the aim to give the legal status to the pathologists, radiologists, and the PGs of Rog Nidan. Our organization is duly registered under Societies Act of 1860 at Charity Commissioner Officer, Nagpur. Postgraduate students, teachers of Rog Nidan and Vikruti Vigyan can become our member. Ours is the only organization which is fighting for the lawful rights of Rog Nidan PGs. Even the NCISM, that is National Commission for Indian Medicinal System, has also directed State of Maharashtra to consider RVPGA as the stakeholder in the constitution of pathology regulatory body of the state. RVPGA is pursuing this matter with the state as well as central government. Not only this, RVPGA is constantly pursuing in the implementation of NCISM salary structure in all the private Ayurvedic colleges across the nation. All the communication and announcement are regularly uploaded on our website www.rvpga.com. Some reminders about our activities. First is regular conduction of online webinars. Excuse me. And offline seminars both on national and international platform. Hands-on training program for the PGs at our partner NABL accredited pathology laboratory at Mumbai, Pune and Nagpur. Publication of research papers of Nidan students and faculties in our peer-reviewed international journal of diagnostic and research, IGDR. Interested can visit the journal site also, which is www.ijdrindia.com. I'm repeating this, www.ijdrindia.com. Fourth, edited book volume series through our international publication RIPR, which is registered in Government of India's ISBN portal also. Those who want to contribute a book chapter can visit RIPR website www.rbpgaiip.com www.rbpgaiip.com. We do legal fight for the issues of Rog Nidan and Vikruti Vigyan PGs at the appropriate forums. We have our YouTube channel, Facebook page, where you can see our educational activities and other activities also. So thank you. Over to you, sir. Hemant, sir.
Thank you, ma'am. Thank you. Precisely introducing our RVPGA organization. Now I would like to invite Dr. Geeta Sathwane madam to welcome and introduce our today's guest.
Thank you, sir. Am I audible, sir?
Yes.
Yes.
Yes. Yes.
Good afternoon, everyone. I, Dr. Geeta Vishwanath Sathwane. I am Associate Professor in Rog Nidan and Vikruti Vigyan Department in Datta Meghe Ayurvedic Medical College, Vanadongri, Nagpur. I heartily welcomes on behalf of Rognidan and Vikruti Vigyan PG Association to all participants who joined this webinar Nedaniki 2023. First of all, I welcome our chairperson of today's session, Dr. Deepali Amle ma'am. I heartily welcome you, ma'am, and I welcome Dr. Subhash Wage sir, the president of RVPG Association. I welcome you, sir. The topic of today's webinar is the investigative approach to immunology. It's my honor and privilege for me to introduce today's guest speaker, Dr. Mayur Bhosle, sir. I give a brief introduction of sir. Dr. Mayur Bhosle, sir, is a MBBS MD in pathology. Sir has done his post-graduation from Institute Topiwala National Medical College and BYL Nair Charitable Hospital, Mumbai Central, Mumbai, Maharashtra in 2014. The dissertation topic was the cerebrovascular disease in elderly patient and autopsy study. One research project is on his credit, which is on the KVPY, that is the Kishore Vaigyanik Protsahan Yojana in the year 2006, breastfeeding practices in the rural areas. Sir has published various articles in national, international journal. One is a atherosclerosis involving the cerebral and the coronary vessels, a comparative analysis, which is published in the Indian Journal of Pathology and the Oncology in 2016. Sir has completed 1 year internship in post MBBS period. Sir worked as a 6-month resident in the blood bank at the Nair Hospital during MD courses. Sir also worked as a resident doctor in the Department of the Pathology in Topiwala National Medical College and BYL Nair Charitable Hospital, Mumbai for 3 years. Sir is a good teacher and having the experience of the teaching to undergraduate students during his residency at the Department of the Pathology and during the house officer at the Department of the Biochemistry in SRTR Medical College and Hospital, Ambejogai. Sir previously worked as in charge blood bank at St. George Hospital, Fort, Mumbai from June 2014 to September 2015. Sir also worked as a consultant pathologist at ICICI Pathology Laboratory at Dr. Hedgewar Hospital, Aurangabad from October 2015 to March 2017. Sir is currently working as a lab head and director at Detect Diagnostic since March 2017. Sir held various job responsibilities like as a medical officer in charge of regional blood bank at St. George Hospital, Fort Mumbai, where he is responsible for signing of the various ELISA reports of transfusion-transmitted infection. Sir also assure the external quality assurance and internal quality control of test and blood components. Sir also managing and conduct the blood donation drives. Sir, also as a consultant pathologist at a Dr. Hedgewar Hospital, Aurangabad. Sir is in charge of the histopathology section. Sir is a holder of various honors and accolades like NTSC scholarship in 2001. The GRE score is a 1300 out of 1600. The TOEFL score is a 115 out of 120. Sir is selected for a fully funded PhD program in the Department of the Molecular Biology, Microbiology, Biochemistry at a Southern Illinois University in Springfield campus, USA in the year of 2010. Sir has completed training as per ISO and the QMS certificate course for internal auditor. Sir is invited as a guest speaker in the Taji Bhale Blood Bank, Aurangabad for NACO supported training of the blood transfusion officers and technician in quality management system for a blood bank. Sir is also qualified MPSC for post of the assistant professor in 2016. I heartily welcome Dr. Mayur Bhosle sir on behalf of the RVPGA sir.
Thank you so much, ma'am. Thank you for the introduction.
And I would like to request you, sir, please start their presentation, sir. Over to you, sir.
All right. Okay. So thank you so much, ma'am, for such a long introduction. Even while listening to the introduction, I could recall my entire journey right from my MBBS up till now.
It's my
So,
pleasure, sir.
yeah, so thank you very much for the same. So we'll start with today's lecture. I'll just let you know that we'll be talking in English and Marathi. I hear ma'am has joined from Gujarat, but I think Marathi will be okay for her, I guess. And what the geographical location, I think of all the participants generally is from Maharashtra region. So we'll have both the languages, which will help you to understand the topic, right? Okay. So today's topic is investigative approach to immunology. So basically, what are we going to cover in this topic? So first, we'll have a brief introduction about immunology. What is immunology? What are the components of immunology, like antigen and antibodies? Then specific reactions of antigen and antibodies. And then at the end of the presentation, the last few slides, we'll have uh, an introduction to the latest or the what we can say the most cutting edge uses of immunology in diagnostics. So let's start with this interesting topic. So first, what do you mean by immunology? So immunology is the branch of science dealing with the components of the immune system, immunity from disease, the immune response, and immunologic techniques of analysis. So immunology म्हणजे काय तर it's a branch, immuno and logy. So immunos, that is immunity, and logos, that is study. So study of immunity, or whatever the immune system is there in our body, how it responds to foreign antigens, what are the components of immune system, and how that reactions can be utilized. So that is immunology. Now in this particular lecture, we are interested in the last part, right? Immunologic techniques of analysis. So we'll start seeing it. So definition of immunity. What is immunity? The ability of an organism to resist a particular infection or toxin by the action of specific antibodies or sensitized white blood cells. Now this definition is very important. Why it is important? Because we get to know what immunity is exactly in this definition. So what is immunity? Ability of an organism. So it's ability of an organism to resist a particular infection. So note here, every word in definition is important. A particular infection. So immunity is specific for that particular infection or toxin by the action of specific antibodies, right? Specific antibodies or sensitized white blood cells. So what do we come to know from this definition? That immunity or immunology is a very specific science. Specific in the sense, what we can say, we have studied organic chemistry or chemistry in our 12th standard, 11th standard. So all the chemical reactions are highly specific, right? In the similar way, antibody and antigen reactions are very, very specific. That is the takeaway from this definition, which will be utilized in today's lecture. Okay, so specific antibodies or sensitized white blood cells. So here we have two components of the immune system also. Specific antibodies. So antibodies are present in liquid form, that is in the plasma of our blood, right? So that is humoral immunity or humoral component of the immunity. Humor means fluid. So fluidic component is specific antibodies, and other is cell mediated immunity, that is sensitized white blood cells. So humoral and cell mediated हे दोन पार्ट पण या इम्युनिटी मध्ये आपल्या डेफिनेशन मध्ये कव्हर झाले. Right. So now we'll see briefly classification of immunity. So immunity is classified as innate or acquired. We'll go stepwise. Innate म्हणजे काय? Innate means which is inborn, right? Inborn means what? It is present right from the birth. So innate immunity. Now the components of innate immunity are not specific for particular bacteria or virus. Innate immunity is present because we have certain barriers to infection which are non-specific, but yet they help our body to combat the oncoming onslaught of bacteria. Now just imagine whenever we are like even standing in the air or sitting anywhere, or even if we are swimming in the water, we are surrounded by microorganisms, right? There are so many types of microorganisms floating in the air, in the water, in the food we eat. But all these microorganisms, generally 99% of the times, they do not infect us. Why it is so? Because they have certain barriers to entry for entering our body. So such barriers are part of innate immunity. So what are they? Skin, tears, stomach, low pH of the stomach, respiratory cilia, normal flora of the gut. These are all part of innate immunity, which hamper the onslaught of infecting bacteria. They do not let the infecting bacteria or viruses easily enter our body. That is structural part, that is innate immunity. Okay. And now for this particular lecture, what we are interested in, we are interested in the acquired immunity. Acquired म्हणजे काय? जसे शब्दच सांगतात आपल्याला. Innate म्हणजे इनबॉर्न, जी जन्मतःच असते. Acquired म्हणजे काय? की जे बाळ जन्मल्यानंतर the body acquires this immunity because of repeated infections, right? So acquired immunity म्हणजे नंतर आपण मिळवलेली immunity, which is natural and artificial. Acquired चे दोन टाइप, नॅचरल अँड आर्टिफिशियल. मग नॅचरल मध्ये पॅसिव्ह ऍक्टिव्ह, आर्टिफिशियल मध्ये पॅसिव्ह ऍक्टिव्ह. आता ऑब्व्हियसली नॅचरल म्हणजे काय? की जी नैसर्गिक दृष्ट्या आपल्या शरीरामध्ये तयार होते, दॅट इज नॅचरल. आर्टिफिशियल म्हणजे काय? की आपण बाहेरून ती इम्युनिटी प्रोव्हाइड करतो, राईट? आर्टिफिशियल इज मॅनमेड, नॅचरल मीन्स इनबॉर्न बाय बॉडी. मग आता ऍक्टिव्ह आणि पॅसिव्ह म्हणजे काय? तर एकदम सोपं आहे, राईट? ऍक्टिव्ह आणि पॅसिव्ह या दोन्हीतला डिफरन्स कळला आपल्याला की खालचं क्लासिफिकेशन कळून जाईल. ऍक्टिव्ह म्हणजे काय? की ऍक्टिव्ह मीन्स की आपली बॉडीची इम्युन सिस्टीम ऍक्टिव्हली त्या इम्युनिटी मध्ये पार्टिसिपेट करते. म्हणजे काय? तर इम्युन सिस्टीम आपली इधर अँटीबॉडीज शरीरामध्ये प्रोड्यूस करते किंवा सेन्सिटाइज सेल्स आपल्या शरीरामध्ये प्रोड्यूस होतात, दॅट इज ऍक्टिव्ह. ओके. पॅसिव्ह म्हणजे काय? की जी आपण बाहेरून देतो, राईट? एकदम सोपं आहे. सो आता नॅचरल ऍक्टिव्ह, नॅचरल पॅसिव्ह, आर्टिफिशियल ऍक्टिव्ह, आर्टिफिशियल पॅसिव्ह, ह्याचे एक्झाम्पल्स काय असतील? तर नॅचरल ऍक्टिव्ह म्हणजे काय? की ड्युरिंग एक्सपोजर टू द डिसीज अँटीबॉडीज आर प्रोड्यूस. आता फॉर एक्झाम्पल आपण एका डिसीजचं एक्झाम्पल घेतलं, जसं की काय म्हणूया आपण, कोरोना व्हायरस. सो कोरोना व्हायरसचं किंवा कोविडचं इन्फेक्शन आपल्याला झालं की त्याच्यामध्ये व्हॉट हॅपन्स, अवर बॉडी प्रोड्युसेस स्पेसिफिक न्यूट्रलायझिंग अँटीबॉडीज टू दॅट कोविड व्हायरस, दॅट इज नॅचरल ऍक्टिव्ह, राईट? आता नॅचरल पॅसिव्ह म्हणजे काय? तर ह्या अँटीबॉडीज ज्या असतात त्या आईकडून बाळाला भेटतात. आपण बाहेरून देतोय का? नाही. नॅचरलीच म्हणजे नैसर्गिक दृष्ट्या जेव्हा आईकडून बाळाला अँटीबॉडीज पास होतात, ती असते नॅचरल पॅसिव्ह. मग ही कधी येते? तर कोविडचंच एक्झाम्पल आपण कॅरी फॉरवर्ड केलं तर जर आई प्रेग्नंट असताना तिला कोविड झाला आणि आईच्या शरीरात त्या अँटीबॉडीज तयार झाल्या, तर ऑटोमॅटिकली बाय बर्थ त्या बाळाला अँटीबॉडीज प्लासेंटा थ्रू ट्रान्सफर होऊन भेटून जातात. दॅट इज एक्झाम्पल ऑफ नॅचरल पॅसिव्ह इम्युनिटी.
Sir, sorry to interrupt you.
Yeah.
Could you please continue in English or Hindi only, because people from all over India have joined us and
Okay,
they are not
sorry.
getting in Marathi.
Yeah,
Sorry.
that is why I had asked at the start of it, but I cannot see the chats over here. Fine. So I'll continue in English. Right. So what do you mean by natural passive immunity? So natural passive immunity is transmission of antibodies from mother to child. To continue our example of COVID virus, if the mother is infected, for example, with the COVID virus when she is pregnant, then such antibodies which are formed in the mother's body are passed to the child via placenta. That is example of natural passive. So that is why the child, whenever the mother has n number of antibodies in her blood and which can be passed to the placenta, the child acquires these antibodies and has some natural protection at birth to various diseases because of the antibodies he received from the mother. Now these have many diagnostic implications. I'll just share a short example. So just consider a peculiar case. For example, the mother is HIV positive and you want to diagnose the infection in the child, whether the child is HIV positive or not. Now, the funny thing in this is the test which we generally utilize for diagnosing HIV till the third generation rapid kits, they detect the antibodies, right? Now, if such newborn child is tested for HIV infection with antibodies, he'll naturally come positive. His test will come positive. Now, the dilemma is in such case whether the child is positive or not. The reaction is showing that the child is positive, but we want to confirm whether he has HIV infection. Now, there is difference between having antibodies of HIV in your blood and having HIV infection, right? So there is a difference between having virus in your blood and having antibodies in your blood. Now, when you test using routine testing techniques for this newborn who is born to an HIV positive mother, he would have acquired naturally antibodies from the mother, right? So these antibodies will be detected and the child will be falsely labeled as HIV positive. So what happens in this case? In this case, for neonatal period, that is why we recommend that HIV PCR testing should be done, which will detect the viral RNA or viral DNA, right? So these are the implications. That is why we should know such things, why these things are important in diagnostics. So when we test for the virus in the newborn blood, if the virus is absent by PCR, the child is negative, even if the antibody screen is positive. That is the implication for this classification. So everything is linked. Once we understand this classification, once we understand the mechanism, then we can interpret the tests accordingly. So this was just a small example. Now the other part, artificial immunity. So again, artificial is artificial active and artificial passive. Now by now it is very clear what is active and what is passive. Artificial active means what? Very good example is vaccination. So to carry forward,
करून घ्या म्हटलं स्टँडर्ड आणि लॅब पूर्ण सगळीकडे कनेक्टिव्हिटी राहील ना
anyways,
कनेक्टिव्हिटी राहील ना रिपोर्ट चालू
so vaccination
आहे का हो हो
artificial active immunity is the example of that is vaccination, right? So what happens in vaccination? To carry forward our example of COVID, in COVID vaccination, which I guess every one of us has taken, what we do, we in COVID vaccination, we introduce the part of the virus which is immunogenic, that is, it will induce our immune system to produce antibodies. So when such part of the virus, either in the form of recombinant RNA vaccine or a killed virus, is introduced in our body reacts to such virus and produces antibodies and sensitized cells. So that is artificial active immunity, in which we give vaccination to induce an immune response which is protective for later on infections. That is example of artificial active. So vaccination is artificial active. And artificial passive, what is artificial passive? Like natural passive, the antibodies are introduced into the body, but how they are introduced? Via IV route, right? So when is this utilized? And a good example is what we do in case of snake bite. So the toxin which is introduced because of the snake bite is neutralized by giving an antivenom. That is an example of artificial passive antibody, in which we inject the patient of snake bite with immunoglobulins which neutralize the snake venom present in the body. That is artificial passive immunity, right? So natural, artificial, active, passive, everything is clear in these examples. Next, we come to the important component of immune system. What is an antigen? So as we have seen in the definition, and immunity is a reaction between antigen and antibody. So what do we mean by an antigen? So the name antigen, that is Greek anti against genos is genus, is given to organic substances of a colloid structure, proteins and different protein complexes in combination with lipids or polysaccharides, which upon injection into the body are capable of causing the production of antibodies and reacting specifically with them. So for a time being, if we forget the part in the brackets, the antigen, the name antigen is given to organic substances of colloid structure, which upon injection into the body are capable of causing the production of antibodies and reacting specifically with them. So in short, what are antigen? Antigens are foreign substances which when injected in our body or introduced in our body are capable of reacting with the immune system and producing antibodies and reacting specifically with such antibodies. So what is important? Specific. Okay, so a specific antigen reacts with a specific antibody. That means whatever different types of antibodies are produced in our blood, they are highly specific and only react with the specific antigen. This is the basis of our entire diagnostic industry, which works on the principle of immunology. So the antigen is highly specific. Antigen antibody reactions are highly specific. And when such antigens are introduced in the body, they give rise to specific antibodies. So the main properties of antigens are then the ability to cause the production of antibodies, that is antigenicity, and the ability to enter into an interaction with the corresponding antibodies, that is antigenic specificity. So causing production of antibodies and then reacting specifically only with such antibodies. So one antigen, one antibody. That is how much specific this reaction is. So what are the features of molecules that determine antigenicity? Foreignness. Obviously, if our immune system starts reacting with our own tissues, that will give rise to autoimmune diseases. That is another part of another lecture. So foreignness is the most important thing. Antigens are generally all foreign substances. Molecular size. Roughly, the larger the molecular size, the more antigenic that particular molecule is. Third, chemical structural complexity. The more complex the molecule, the more antigenic it is. And fourth, antigenic determinant, that is epitopes. So a specific part of the antigen which reacts with antibodies, that is called an epitope. So as we can see over here, the antigen is quite big, but there is only a specific part, such as, for example, this spike, which reacts with the specific antibody. This is called epitope. This is called antibody, and the part of the antibody which reacts with the epitope is called paratope or receptor site, right? This is a very important part for understanding. That is why I have taken this. Now, the antibody specifically reacts with the epitope, right? This is a bigger antigen, and this is the epitope, which is specific part which reacts with the antibodies, and the antibody molecules are attached to this, right? So the green will react with green, the red will react with red, and the blue will blue. This determines the specificity of the reaction. Now there is a thing called hapten. What is an hapten? Hapten is a molecule that cannot induce an immune response by itself, but can react with specific antibody. Okay, so haptens, for our understanding, haptens are half antigens, half antigen, hapten. So haptens are half antigens which by themselves are non-immunogenic, but when combined with different substances can become an antigen and induce an immune response. This is important because in some reactions, for example, allergy to penicillin. So penicillin by itself is non-allergic. Penicillin acts as a hapten, which when combined with proteins in our body becomes antigenic. That is one of the examples why we should keep haptens in mind. Now we are clear with antigens. Now we'll see what is an antibody. So an antibody or an immunoglobulin is a specialized immune protein, a highly specialized immune protein. Why immune protein? Because it is produced by immune system. So a specialized immune protein or a specialized protein produced by our immune system, produced because of the introduction of antigen into the body. So what is an antibody? A specific protein which is produced in our body because of the activation of the immune system and because of the introduction of a specific antigen, which possesses the remarkable ability to combine with the very antigen that triggered its production. So it is a highly remarkable antibody or highly remarkable molecule which is produced because of an introduction of antigen, and it can specifically react with that particular antigen. So this is the scanning electron micrograph of an immunoglobulin. And I find this very, very fascinating. That is why I've shared this photograph, because see, this visual impression gives you of gives you an idea that the things are what we can say, how they look exactly and how we use them for representational purposes. In next slide, you will see that we represented it as a Y shape. But when we see over here, this tells us that how beautiful it looks at actual. When we draw a caricature or when we draw a very schematic diagram, we miss this beauty of nature. That is why I have shared this photograph. Right. So it is a Y-shaped molecule. Now, in next slide, we'll see the parts of this Y-shaped molecule, which are very important to understand our diagnostic techniques. So this is a schematic of that diagram. So just see how beautiful this is and how ugly this is when we reduce it to a schematic, right? So this is a Y-shaped molecule, and antibody is a Y-shaped molecule, which has these light chains and heavy chains. So this small part is the light chain, and this larger part, which has a bend, that is a heavy chain. Now you'll see over here, every light chain and heavy chain has a variable part, V and C part. C is the constant part, V is the variable part. Now, why it should be a variable part? Because as we have seen, a particular antigen only reacts with a particular antibody. So every antibody has a variable part which is designed according to the antigen with which it will react. And it has a constant part which will trigger the immune response. Because the cells, the receptors on the other cells to which antibodies attaches, the immune cells, these are constant, which recognize this part of the antibody, and this part, variable part, attaches to the antigen. Now, why this is diagnostically important, we will see later on, but I'll just share in short over here. The antigen attaches to this part, and the specificity of this part is utilized in the secondary antibody, which attaches to this part and triggers an reaction which we measure electronically, right? So this part, the constant part, is important for biological activity of the antibody, and this variable part is responsible for the antigen binding. So what do we mean by biological activity of the antibody? Whatever that antibody does for our immune system. Means what? Triggering the immune response, triggering the cells, alerting the cells, or alerting other lymph nodes to produce more cells which recognize this antibody, or alerting the mast cells and triggering the allergen, or what we can say histamine release, which is responsible for our allergic reactions. So this part, constant part, is responsible for biological activity, and the variable part attaches to antigen. Now it is very clear why it has to be variable, because antigens are variable, or antigens, as the number of antigens which are exposed in our body increases, the variety of these antibodies increases. Now, what are the different immunoglobulin classes? So these are the different immunoglobulin classes. Just a short introduction over here. We have IgG, immunoglobulin G, IgM, immunoglobulin M. Okay, then IgA, IgD, and IgE. So these are different classes of immunoglobulins. And just in short about that, what are their biological functions? Right. So this is the monomer. Means what? It's just one molecule. This is the pentamer. That means it has 5 antibodies linked in a single molecule. This is a dimer with 2 molecules, and these again are monomers. So immunoglobulin G, what is it? Is enhances phagocytosis and protects fetus and newborn. Now this is very important, right? So again, the example of HIV, which we had discussed earlier, when the mother passes her antibodies to the child, they are of only IgG class. Now, why it is so? Because as we see over here, it's a monomer. It has a less molecular weight, and that is why only IgG type of antibodies are capable of crossing the placenta. Right. Now you can say, sir, so in the HIV case, what we should do? If we check for IgG in the fetus or the child, specifically IgG will come positive, but IgM will come negative. Why? Because IgM is pentamer, it does not cross placenta, and if IgM is detected in the child or the fetus, that means his immune system is triggered. Now, when will the immune system will be triggered? If it is exposed to the antigen. That means the child's body has the neonate's body has the virus. That is why IgM is produced. So whenever IgM is produced, that means there is infection, and if IgG is detected in the child, that means generally it has been passed from the mother. That is why understanding this structure is important. So later on, there does not remain any confusion, right? So these are the specific examples or different types of antibodies. Now this is specifically for IgA. Immunoglobulin A is the only antibody which is found in our secretions. Nasal secretions, GI secretions, IgA is present, right? So आता ह्याचं importance, why this is important? Because do you know why there was interest in developing a nasal spray for COVID vaccine? This will give you the answer for it. Because when you give a vaccine by intravenous or intramuscular route, it does not trigger the mucosa. So what happens? That particular person is protected from the virus, but the virus can colonize his mucosa and be transmitted to others. So when you see this diagram, you quickly understand why the natural route of infection for vaccine is important. That is why oral polio vaccine is more what we can say effective in preventing the spread of the polio virus rather than IPV or intramuscular vaccine, which was used injectable vaccine previously. What happens when OPV is given? It stimulates the gut mucosa to produce immunoglobulin A, which lines the gut mucosa and prevents the viruses from entering the feces and transmitting elsewhere. That is why IgA is very important in mucosal infections like nasal mucosa, gut mucosa, or wherever there is mucosa in our body. IgA is the secretory immunoglobulin which is present in such mucosa. Right. So we'll come to now antigen antibody reaction. What is an antigen antibody reaction? Antigen and antibodies combine with each other specifically and in an observable manner. So specifically and in an observable manner. Why we are interested as diagnosticians? Because these reactions can be observed. And there are various techniques for observing these reactions, which we are now going to study. So what is serological reaction? Antigen antibody reactions in vitro are called serological reactions. So everywhere we see serology lab or serological testing. What do we mean by that? When antigen and antibody reactions in vitro, right? In vitro manje kai? In vitro means what? In the petri dish or in the laboratory. So whenever antigen antibody reactions are there in the laboratory, those are called as serological reactions. Uses in vivo, that means in the body. It is based antigen antibody reactions in the body are basis of antibody mediated immunity in infectious diseases, as we have seen. And other example is tissue injury in hypersensitivity and autoimmune diseases. So when our own immune system reacts with our own antigens, that gives rise to hypersensitivity or autoimmune diseases. That is the example of in vivo antigen antibody reactions. In vivo means in body. In vitro, what are the examples of antigen antibody reactions? Diagnosis of infections in the laboratory, in epidemiological surveys. For example, if you want to know how many people are infected with COVID, then what we will do, we'll get the vaccination history. Those who are not vaccinated but who become antibody positive are infected with the virus. So in epidemiological surveys, then detection of non-infectious and agents such as enzymes. Right. So various enzymes in our body are also biochemically detected by using antigen antibody reactions. Those reagents which we use in our labs, many of the times those are antibodies against the enzymes. Then detection and quantification of either antigen or antibody. So detection and quantification. So either antigen or antibody. This example we can say, for example, very common example of detecting thyroid stimulating hormone, TSH. So when TSH is used as an antigen, it reacts with a specific antibody, which can be this reaction can be used for quantifying how much TSH is present in the patient's body. So these are different examples where antigen antibody reactions are used. We are interested in vitro part, right? So diagnosis of infections, then detection and quantification of either antigen or antibody. These are the important parts for us as diagnosticians. So general characters of this reaction, that is, the reactions are very specific. Entire molecules react. There is no denaturation of antigen or antibody. That means what? At the end of the reaction, you can separate the antigen and antibody. There will be no destruction of the structure. Combination occurs on surface. Combination is firm but reversible. The combination of antigen and antibody is firm, but you can reverse the reaction. Affinity, intensity of attraction of antigen or antibody is called affinity. And avidity is strength of the bond formed after formation of antigen antibody complexes. Okay, both antigen and antibody participate in the reaction, and antigen and antibodies combine in varying proportions. Okay, the proportions of their combinations are variant because antibodies generally are bivalent or pentavalent, as we have seen, and antigens are multivalent. So why this is important? A single antigen can attach to multiple antibodies. That is what we should keep in mind. So this is just a schematic of which reactions or which forces are involved in antigen antibody reactions, just for informative purposes, which we have learned already in our 12th standard. Van der Waals interactions, ionic bonds, hydrophobic interaction, hydrogen bonds, ionic bonds. So as we know, ionic bond are the most strong of these all four forces. Now, first example, precipitation reaction. So what is a precipitation reaction? When a soluble antigen combines with its specific antibody in the presence of electrolyte, that is sodium and chloride, at suitable temperature and pH, the antigen antibody complex forms insoluble precipitates. So the most important thing in precipitation reaction is a soluble antigen is used. Okay, it reacts with specific antibody in the presence of electrolytes, that is in appropriate environmental conditions. That is, there has to be presence of electrolyte which enhance this reaction, and at a suitable temperature and pH, the antigen antibody complex forms insoluble precipitates. So a soluble antigen is converted into an insoluble precipitate. Now see here, observable manner. So there is a specific, the antigen antibody reactions are specific and they react in an observable manner. So here we can observe the formation of precipitates. Soluble antigen converted into precipitates, that is insoluble antigen, that is called as a precipitation reaction. Okay, now there is a variation of this. When the antigen antibody complex, instead of sedimenting, remains suspended as the floccules, the reaction is known as flocculation. Right. So when instead of forming a precipitate sinks at the bottom, and flocculation means what? The complex floats on the surface, that is called flocculation. So flocculation is a variant of precipitation, and a good example of this is the VDRL test. The slide VDRL test, not the rapid card one, the slide VDRL test, which was the original form, which is till today used in many of the blood banks for checking the syphilis infection in the donors, is an example of flocculation reaction. If most of you have seen this reaction, you will remember that what happens, the dark particles which are scattered, they come together, they form a complex, and such complex is seen floating on the surface of that tile. So that is flocculation for you. Now this zone phenomena, which is seen both in precipitation and agglutination reaction, is very important for us as pathologists. So what is zone? So zone phenomena is seen, okay, when antibody or antigen is in excess and suboptimal immune complexes are formed. Now these three terms are very important: prozone, zone of equivalence, and post zone. We'll just see the diagram for this, which will make you understand. So what happens whenever there is an excess antibody in the blood, and when such serum is used in the testing, and if the antigens which are added for checking such antibodies are not put in optimal quantity, or the antibody is not diluted, what will happen? Even if in the first part, if the antibody is in excess and the antigen is less, you will get a false negative reaction. Right. That is why it is important to check the precipitation reaction in dilution. Right. Because what happens, sometimes the patient has in particular infection, but the test come negative. In such case, this prozone phenomena is responsible for such negative tests. So when we dilute the sample, what we do, we reduce the amount of antibody, right? Or if we put excess antigen, then again we reduce or we disturb this equilibrium and let the reaction come to the zone of equivalence, and then the precipitates are formed. So that is why zone phenomena is very important. Mechanism of precipitation reaction is lattice hypothesis, in which a soluble antigen, this antigen combines with different antibodies, which is the antigen by itself is soluble, but the complex which is formed, that is insoluble and it precipitates. So that is the mechanism of lattice hypothesis. Application of precipitation reaction. Precipitation tests are mostly used to detect antigen. One example which I gave you, that is VDRL. Another is the Vidal test. Vidal test is also a type of precipitation reaction. As less as 1 nanogram of protein can be detected, it is less sensitive to detect antibodies. Precipitation test may be used for identification of blood and seminal fluids also. Precipitation in liquid example is, as we already discussed, VDRL, and then ring test for detection of C-reactive protein, that is CRP test, then Lancefield grouping of streptococci, and Ascoli's thermoprecipitation test. These two tests, Lancefield and Ascoli's, they have only historical purpose. They are generally not used, but CRP is used. The immunoturbidimetry principle, which is used in biochemistry, is also a very good example of precipitation reaction. Immunoturbidimetry means what? Immunological reactions are used for measuring the turbidity which is found, which happens into the solution because of the presence of particular antigen of interest. That is immunoturbidimetry. Okay, that reaction is used for specific detection of what we can say, the assay or the particle of interest, that is called the turbidimetry assay, right? So immunoturbidimetry is used for detection of light chains, heavy chains, etc., right? So immuno and turbidimetry, even it is used in very what we can say, the routine measurement of HbA1c or ceruloplasmin. You must have heard about technique called nephelometry. So nephelometry is a variant of immunoturbidimetry, in which this complex is measured by passing light through it, and the intensity of the absorbed light is equal to the concentration of that particular antigen in the solution, which has become turbid because of using specific antibodies. That is immunoturbidimetry, right? So this is just an example of ring test. Zone of equivalence, the ring is formed, antigens soluble become insoluble, right? So soluble antigens become insoluble in zone of equivalence. That is why here we don't get a ring, here we don't get a ring, and only at the specific point we get a ring. Precipitation in gel, right? So radial immunodiffusion, that is again an example of using precipitation reaction in gel. This again is for research purposes only, not used generally in the diagnostic lab, but we should know this technique, in which agarose gel is impregnated with either antigen or antibody, and then a well is dug in it, and we put an antigen if we want to measure the antigen, we put an antibody if we want to measure the antibody, and whenever there is zone of equivalence, there is precipitation reaction. The antigen antibody react and a ring is formed. Now the thickness of this ring is generally proportional to the concentration of the antigen or antibody, whatever we are measuring. Always remember, whatever we want to measure is put into the well, and whatever specific antigen or antibody against that antigen, that is put into the agar. So when we put serum into the well and we want to measure antibodies in that serum for a particular disease, the antigen for that antibody is impregnated into the agar, and antibody or serum is put into the well, right? So this is radial immunodiffusion. Immunoelectrophoresis. So as the name suggests, immunoelectrophoresis. Phoresis means separation, electro means using electrical forces, and immuno means using immunological reaction. So as in radial immunodiffusion, here the antigen and antibody they diffuse because of the gravitational forces or the capillary forces. But what we do in immunoelectrophoresis, we apply current. So applying positive and negative electrodes spreads these antibodies very fast, or increases the what we can say, increases the intensity of the reaction by moving those antigen and antibodies fast. So immunoelectrophoresis is against a variation of radial immunodiffusion. Now agglutination reaction. Agglutination, understanding agglutination is very important. Why it is important? Because it is the basis of many laboratory reactions. Our routine test, common blood grouping, right? Blood grouping is a type of agglutination reaction. Direct Coombs test is a type of agglutination reaction. Indirect Coombs test is a type of agglutination reaction. Then again, hemagglutination reactions, which were used previously for identification of various viruses or virus infection, are again a type of agglutination reaction. So what is agglutination reaction? When a particulate antigen. Now please remember this, in precipitation, the antigen was soluble. In agglutination, the antigen is particulate. When a particulate antigen combined with its homologous antibody in the presence of an electrolyte at suitable temperature and pH, the particles are clumped or agglutinated. So a particulate antigen reacts with antibodies, and that particulate antigens are clumped together or agglutinated. That is agglutination reaction. Right. So agglutination reactions are very sensitive, relatively easy to read. Why it is easy to read? Because we can see with naked eye the agglutination. Right. Blood group, when we do the blood group testing, whenever we put the antisera, we can easily see with our naked eye that agglutination has formed. Okay. Then agglutination occurs optimally when antigen antibodies react in equivalent proportion. Again, zone phenomena can also be seen over here. Right. So mechanism is same, lattice hypothesis. See over here. So if we see this as an example of our routine blood group testing, these are antigens, what we can say A antigen, B antigen, etc. on the RBCs, and these are specific antibodies which we use, which when the antisera is put on those RBCs, this IgM type of antibody. Now IgM is important. See, IgG cannot be used over here because IgG is monovalent. It will combine only with this thing. It will not cross combine with other antigen or for clumping. So only IgM can give rise to agglutination reaction, right? So this antibody links these antigens together and forms a complex. Okay, and they are detected, right? So an agglutination reaction occurs when antibodies react with antigenic determinant sites on antigen carried on neighboring cells, such as these bacteria or red blood cells. The particulate antigens agglutinate. So this particular antigens agglutinate means what? They are clumped together. IgM is the most efficient immunoglobulin for agglutination, is shown here, but IgG may also participate. However, it is not that efficient. Application of agglutination reaction, very important, direct agglutination test, or how it is used. So direct agglutination test detects antibodies against relatively large cellular antigens such as those on the red blood cells, bacteria, and fungi. Slide agglutination, very good example. Blood grouping. Okay, then bacterial agglutination, right. Tube agglutination. So whenever agglutination forms in a tube, that is called agglutination reaction. Whenever the blood grouping is done using tube technique, again, that is an example of agglutination reaction. Other example, enteric fever, brucellosis, typhus fever, infectious mononucleosis, etc. These can be detected using agglutination reactions. Now indirect. Direct means was direct, indirect, either the RBCs or the bacteria directly react with the antibody. In indirect, what happens? So here individual antigens, they are small, they are adsorbed in on other particles. So other bigger particle is utilized as a support medium for attaching this antigen. In simple words, that is indirect agglutination reactions. Okay. And these are known as lattice agglutination tests, commonly used for rapid detection of serum antibodies against bacteria and viruses. Okay, ASO titer. ASO titer is also an example of agglutination reaction. In such indirect passive agglutination tests, the antibody reacts with soluble antigen adhering to the particles. Okay, they agglutinate with each other. Now, here I'd say earlier the antigen directly used to react with the antibody. What happened over here? Either the antibody or the antigen is adsorbed on a surface of another larger particle, and then they react with each other. That is called indirect or passive agglutination. Okay. Application hemagglutination, as we have seen, for detection of blood grouping. Coombs test, which is again very important test used in the detection of hemolytic anemia, is a type of agglutination reaction. Another example is hemagglutination test. As I have said, it is only for historical purposes, which was used for detection of viruses. So some viruses, because of their intrinsic properties, can agglutinate RBCs. So presence of such viruses can be detected by incubating them with RBCs, and if the RBCs agglutinate, that virus is present. If they don't agglutinate, the virus is absent. Such was the simple principle of this test. And this is important to understand Coombs antiglobulin test. So how it is done? Coombs agglutination test, what we do, the antibodies react with the RBCs. So these are antibodies which are responsible for autoimmune hemolysis. These antibodies already are coated with, the RBCs are already coated with these antibodies. Now this A part, which is mentioned over here, these two reactions till the antigen antibody complex formation takes place in the patient's body. And this part, the B part, takes place in the laboratory. So when such blood from the patient is taken and incubated with antibodies which react with these antibodies. Now, as we have seen this Y part, so what we see over here, the Coombs reagent. What is Coombs reagent? Coombs reagent is an antibody against this Y part of the antibody, right? So the antibody which is responsible for hemolysis, this part, this yellow, this blue part, this blue antibody is detected using this green antibodies. So such RBCs, when react with these antibodies which are coated, they become clumped, right? They become clumped, and we get a positive reaction. That is Coombs reagent, Coombs reaction, right? Now, the most important part, I am skipping the historical parts. Now, the most interesting part we are starting over here, the latest techniques in which we use immunological reaction. Now, the most important and very fascinating techniques is immunofluorescence. So as we see over here, immuno and fluorescence. What do you mean by fluorescence? If you understand this word, fluorescence means producing light. So a reaction which uses immunological principles and produces a light, that is called as an immunofluorescence reaction. So what is the principle of immunofluorescence? Fluorescent dyes, okay, can be covalently attached to the antibody molecules and made visible by ultraviolet light in the fluorescent microscope. So this technique is used very commonly for detection of anti-nuclear antibodies. ANA, IFA, wherever we say IFA, that is immunofluorescence. So IFA technique, immunofluorescence technique, is used in the detection of anti-nuclear antibodies in the lab, which we also do in our lab. The technique is very fascinating. What happens? As we have seen, a particular antigen reacts with antibodies. So in anti-nuclear antibody, ANA, that is the full form of ANA, anti-nuclear antibody. So what happens there? We use different antibodies against the component of nucleus. And such antibodies are labeled with a dye, which when viewed under the ultraviolet light, it glows, right? It gives rise to visible light. That is the principle. So in ANA testing, immunofluorescence, what we do, we take the patient's serum, incubate it on the cells which are taken from the manufacturer, and that antibodies, if present into the serum of the patient, they are attached to these anti-nuclear antigens. And when this tile is viewed under the ultraviolet light, it gives rise to a visible light that is seen using a microscope. That is the principle of immunofluorescence, right? So how this works? Direct immunofluorescence. So this is the cell surface, this is the antigen, this is the antibody which is labeled with a fluorescent dye. Okay, in its simplest form, it is used for identification of bacteria, viruses, or any other antigens which we can confirm visually under the ultraviolet light using a microscope. Right. It is also used in the diagnosis of rabies, in which biopsy is taken, either a corneal scraping or in rare cases, brain biopsy is also taken and tested for rabies virus using a antibody which is specific against the rabies virus and which is labeled with a dye. Indirect immunofluorescence. So always remember direct and indirect. What is the difference? Direct means the antibody which reacts with the antigen is labeled with the dye or a color producing mechanism, that is direct. And in indirect, what happens? Another antibody which attaches to this antibody is used, right? That is only the difference between direct and indirect, which we will also see further in other techniques. Now, this is fluorescence. Next is ELISA, the most important technique. What is the full form of ELISA? Enzyme-linked immunosorbent assay. Enzyme-linked immunosorbent assay. This gives us the principle. The name itself gives us the principle. How it is used? Enzyme, right? So what enzyme is used? Enzyme is used for producing color into the reaction. So enzyme-linked immunosorbent. Immuno, again, what by immuno what we mean? Immunological reaction. And sorbent means what? That particular antigen or antibody is absorbed at the bottom of the well. So enzyme linked immunosorbent assay. Assay means what? Measurement. Whenever the word we use assay, we use it for measurement. So what is ELISA? Enzyme linked immunosorbent assay is so named because the technique involves the use of an immunosorbent, that is an adsorbing material specific for one of the components of the reaction, the antigen or antibody. So there are two basic methods. They are direct or sandwich ELISA, which can detect and quantify antigen, and indirect ELISA, which can detect and quantify antibody. So ELISA is so named because technique involves the use of an immunosorbent absorbing material specific for one of the component, that is antigen or antibody. As an integral part of healthcare, in vitro diagnostic enables the healthcare professional to screen, diagnose, treat, and monitor and manage the diseases using ELISA. A wider range of ELISA, the most widely used IVD assay in clinical diagnosis, have been developed. Their deployment has contributed to considerable advances in healthcare diagnostics. So we'll see how ELISA works. So schematic of direct or indirect ELISA. How does it work? As I have said, direct means what? Always the antibody which reacts with the antigen is labeled. Okay. Now what this label is? It's a streptavidin molecule, which when incubated with the enzyme substrate gives rise to a color, generally a yellow color. So as we have seen, the technique is very, very basic. If we understand it clearly, what we are using an antigen and antibody, right? This reaction, the antibody is labeled with a mechanism which can produce color, and this color can be measured using photometric reaction. That is the basis of ELISA. And indirect, again, as I have said previously, what happens in indirect? The second antibody is used, that is called a secondary antibody, which reacts with the primary antibody and gives rise to color. Now this is the adsorbent material, right? And this is the antigen of interest. So this adsorbent material is coated with antibodies. This is the antigen. Now these antibodies capture this antigen, and again a second antibody is put which reacts with this antigen, and a third antibody, that is the label antibody, is put which reacts with this antibody, and then we put a color producing substance which reacts with the enzyme present on this secondary antibody to give rise to a color, right? So this color is produced, which is measured using the ELISA reader, which gives us the understanding and the quantification of either antigen or antibody. So this is direct and this is indirect ELISA. We'll see a video which will clear our doubts of all the ELISA questions. So let's start with this video.
ELISA is short for enzyme-linked immunosorbent assay. With this plate-based technique, we can detect and quantify soluble substances such as proteins. During an ELISA, the antigen, which is the target molecule, is immobilized and specifically detected by antibodies, which are enzyme-coupled. When providing a colorless substrate, the enzyme produces a measurable, colorful product. An antibody is attached to the bottom of the well. This antibody is specifically designed for binding a specific protein of interest. Another soluble antibody is provided. The antibody is also specifically covering protein X. This antibody is conjugated to an enzyme. When we now add a substrate, the enzyme can convert it to a colorful product, which can be measured later. There are many variants of an ELISA. In the direct ELISA, the antigen is immobilized on the bottom of the plate, and the enzyme-linked antibody directly binds to the antigen. In the indirect ELISA, a primary antibody binds our antigen, and then a secondary antibody binds the primary antibody. The secondary antibody is enzyme-linked. The most common type is the sandwich ELISA. A primary antibody is immobilized and captures the antigen of a solution, and another primary antibody specifically binds the same antigen. But all three ELISAs have one thing in common. The last antibody binding is always conjugated to the enzyme which confirms binding. Here we provide an example of an ELISA. For this sandwich ELISA, we have two wells, both coated with primary antibody specific for protein X. On the left, we provide a sample containing our protein. On the right, we have the control without the target protein.
That's a
Our
negative
antigen
control.
in orange will be immobilized. A washing step will discard everything else. Now we provide the enzyme-linked antibody. It will bind our antigen in the left well and remains bound after washing, whereas the antibody is discarded in the control. In the last step, a colorless substrate is given. Since the enzyme, in most cases horseradish peroxidase, is exclusively present in the left well, it produces a measurable, colorful product here. This can be investigated and also quantified using spectrophotometry. The control remains colorless since substrate is not converted here. If you want to have a closer look into the direct or the indirect ELISA, you may click one of these videos here.
All right, so that was ELISA for you. Very easy to understand if we get the basic principles, right? So why it is important? Because it is the most common methodology used in the labs for detection of various antibodies. As you all know, right now we are facing what we can say a great increase in the dengue infection, and dengue ELISA is the most basic test which is used in the laboratories for detection of dengue. There are also rapid card tests, but ELISA is very specific. And as a diagnosticians, we should understand in ELISA, we should always have a positive and negative control. Why? Because the negative control will tell us that we are getting a specific reaction. We are not getting a non-specific reaction. And the positive control will tell us that our reagents are working perfectly. That are the uses of positive and negative control. Right. So that was ELISA for you. Now, another important technique, which is CLIA. Now, many of you must have heard about this technique. If you are working in labs or seeing modern labs, all the modern labs have instruments for measuring hormones, tumor markers, etc. Now, for measuring these hormones and tumor markers, the technique which is used is CLIA. CLIA is very similar to ELISA, but differs in slightly different aspects, and that in what aspects it is different from ELISA, that we'll check. CLIA. So what is the full form of CLIA? Chemiluminescence immunoassay. Now again, we'll just stop over here for a minute. what do you mean by chemiluminescence? Chemi means what? A chemical reaction which produces luminescence, that is light, using immunological principles, and then such light is measured, that is assayed. So chemiluminescence immunoassay, a chemical reaction which produces light, is utilized for detection of particular antigen bound antibody bound antigen, and it's used for measuring such antigen in the blood, that is chemiluminescence immunoassay. So what is the principle? In the presence of complementary antigen and antibody, the paratope, that is the part of the antibody which reacts with the epitope, the paratope of the antibody binds to the epitope of the antigen to form an antigen antibody or an immune complex. So this first part takes care of the immuno part, that is the immunological reaction. Okay. Then what happens? Estimating the levels of such immune complex by use of labeled antibodies form the basis of CLIA. So whenever we estimate the presence of such immune complex by using chemical reactions, that is CLIA, or that forms the basis of chemiluminescence immunoassay. It involves use of stationary solid particles coated either with the antigen or antibody of interest. Okay, so solid particles are coated with the antibody of interest, and then a patient serum is put. Post incubation with the patient serum, it ensures that intact immune complexes are formed, and then a substrate is added. So what does the substrate do? The substrate reacts with the secondary antibody, which is labeled with an enzyme which can produce color, that is principle of ELISA, and in this case produce light, that is principle of CLIA. This results in generation of light, the intensity of which is directly proportional to the amount of labeled complexes present, and which indirectly aids in the quantification of the analyte of interest. Means what happens in ELISA? A color is produced, it's a colorimetric reaction, and here luminescence or light is produced. That is the only difference between ELISA and CLIA. And another difference is CLIA can be used in liquid form, while ELISA you have to use the adsorbent surface. So we'll see how CLIA works. The intensity of the light is then measured in terms of relative light units. Now, when we plot the intensity of light against the concentration of our antigen of interest, we get a graph that is called calibration of the instrument or calibration graph. When we put a patient's serum and then measure the light produced because of the analyte of interest against such graph, we get the quantity of that antigen of interest in the patient's serum. That is the principle of CLIA. So the main advantage of this technology includes sensitivity and its ability to be unaffected by the background signals. Why unaffected? Because whatever the color of the sample, we don't have to do. Even hemolyzed samples can be used because we don't measure the color, we measure the light produced. Also, the analyzers working under this principle are simple in design and operation. So we'll see a short video about CLIA, how it works. So a magnetic particles with streptavidin capture the biotin labeled antibody. So this is a labeled antibody. It is captured by these magnetic particles. Right. Then we put a sample. This specific antibody then reacts with this. For example, TSH molecule from the patient's blood. This TSH molecule is captured on this antibody. Then the acridium ester labeled secondary antibody is put into this, this liquid. It combines with the antigen. Now this is the light triggering mechanism. Then a substrate is added. So this enzyme reacts with the substrate to produce light. And then this light is measured into the instrument. That is, in essence, principle of CLIA. So that was CLIA for you. I'll explain in 5 minutes immunohistochemistry, and then we can take questions. Now, this again is a very, very important part which is used into the modern laboratories for detecting various cancers and prognosis of those cancers based on various markers which can be produced, which are present in the tumor and which produce a specific reaction by using immunohistochemistry techniques. What is IHC? Immuno, that is immunological principles are used, antigen antibody reactions are used. Histo means on tissues or histology, and chemistry or biochemical reactions. So immunological reaction, that is antigen antibody specificity, is used onto the tissues, and how it is visualized by using biochemistry or biochemical reaction. That is in principle immunohistochemistry. So immunohistochemistry is the application of a labeled or enzyme-bound antibody to identify a specific antigen on histological sections, which is visualized under light microscopy by means of a color signal. So what happens in immunohistochemistry? A specific labeled antibody, enzyme-bound antibody, is used for reacting to a specific antigen on the tissues, and then a substrate is put. So if that antibody bounds to a specific antigen, that is the antigen of interest is present on that tumor, then such antibody will bind to that antigen. If it is absent, it will not bind to the antigen. So it will give a negative reaction. If present, it will bind to the antigen, and then we'll put a substrate which will produce a color. That is the principle of IHC. So direct method, again direct and indirect, is to apply labeled antibody directly to the tissue sections. The marker then demonstrates the cellular location of the antigen in the section. So a tissue section, labeled direct primary antibody and marker, that is direct IHC. Then again indirect IHC, again whenever indirect is there, we use a secondary antibody. So primary antibody reacts with antigen, secondary antibody reacts with the constant region of that antibody, and it is labeled with a marker. A substrate is put and it produces color, which can be seen under the microscope. That is immunohistochemistry. So we'll see a short video how IHC works.
Immunohistochemistry. Immunohistochemistry includes in situ detection of antigens in tissue sections and cells, using monoclonal and polyclonal antibodies. Detection is through visualization of the antigen using microscope.
Immuno, that is antigen antibody based. Histo, that is tissue based. Chemistry, that means the chemical reaction.
Protocol for IHC staining.
So what we do in IHC staining, there is a tissue which is paraffin embedded. Then we do deparaffinization or rehydration of the tissue. We do antigen retrieval, that is we get antigen which are denatured because of formalin. They are retrieved using various techniques. Then we use a blocking technique for blocking inherent antibodies into the tissues. We don't want a non-specific reaction. Then a power block, which again neutralizes all the antibodies. Then we put a primary antibody which attaches to the antigen from the tissue. Then we rinse. Every time you see in ELISA or IHC, we always rinse to remove non-specific reactions. Then we put a secondary antibody which attaches to the primary antibody. Then we put a chromogen or a substrate. Again, see, every step is followed by washing. Put a substrate which reacts with the secondary antibody, which is bound to an enzyme, produces color. And then we put a counter stain which gives a background stain which helps us identify the reaction. And then we mount this slide and see it under the microscope. How beautiful IHC looks, and the beauty is you can use different colors for different antigens. So this is a schematic of the reaction which we saw. Tissue, antigen, primary antibody, then a secondary antibody which is biotinylated. Then labeled enzyme conjugate, that is streptavidin, and substrate. So a secondary polymer is used for enhanced reaction. That is super sensitive because multiple secondary antibodies are combined in a polymer and they react to give a highly intense light or a highly intense color. So that is the beauty of ISC, which we can, even on single tissue, we can use different color antibodies to highlight different antigens. Because every color is specific for that particular antibody. And then using a secondary antibody in the form of polymer enhances the reaction because multiple antibodies are attached together to a single polymer. That is how ISC works. So that's it in today's presentation. Thank you everyone for listening patiently. And please, if you have any questions, we can take a few questions with the what we can say permission of the organizers, or the organizers can take it forward from here.
Please ask.
I'm just telling you, this can have only two connotations. Either everybody has understood everything or nobody has understood anything. The problem with online presentation is that I don't get to see the faces of people sitting over there and listening to the lectures. So if I get to see that in a classroom, I can understand that if it is going potential or people are understanding, and then I can modify accordingly. But that is the limitation of the technique which we are using. So much possible.
I hope our PG students would have take a lot of benefit from this lecture, sir.
Yeah, same from here. But that is what I said. I really like to present it in a classroom, but unfortunately, that is not possible. And the other thing was, it is a very what we can say, a very vast and complex topic, which had to be fitted in what we say a time of 1 hour.
Thank you, sir. Thank you very much. It was very informative and elaborative lecture on investigative approach to immunology. Thank you, sir. After this, I invite
Hemant, there is one question.
Yeah, multiplex IFC.
Yes, sir. Please.
Okay. So as I said, Dr. Prachi, in multiplex ISC, what we do is different type of antigens can be visualized in a single tissue. So as I said, different labeled, different colored antibodies can be used to target particular antigen. Say, for example, a blue antibody for a particular antigen, then a red antibody for another, then a dark green for another. That is called as a multiplex ISC, in which different antigens are detected simultaneously. Antigen detected simultaneously. And there is another also a variant of multiplex, not exactly multiplex, but you can say tissue microarray technique for using ISC, in which what we do is, in order to save the antibodies, we take different, what we can say, for example, if you want to detect a particular, let's say an example of estrogen receptor in the breast cancer. So ERPR needs to be detected in breast cancer. We want to save the antibody, and there are, say, 5 patients or 5 samples which need to be run at a time. So what we do is, we take a small course of tissue from all the 5 different patient samples, then put it on a slide, single slide, 1 to 5, label it accordingly, what is 1 which patient, 2 is which patient, until 5 which is patient, put a positive and a negative control against that, and then use a single reaction to identify ERPR status of all these tumors. That is called the tissue microarray technique also. So multiplex ISC and tissue microarray techniques, these are variations of ISC which are developed for different what we can say utilization of a technique, right? I hope this answers your question.
Thank you, sir. After this lecture, I would like to invite today's chairperson, Dr. Deepali Amble madam, for chairperson remarks.
Thank you, Hemant sir. Good afternoon, everyone. Am I audible?
Yes, ma'am.
Okay. Thank you, first of all, Vague sir, President of RVPGA, for inviting me as a chairperson in this our Naidanik 2023 webinar series organized by Rognidan Vikruti Vigyan PG Association. Sir, it's a great honor for me to have the opportunity to narrate the speech delivered by our eminent speaker, Dr. Mayur Bhosle sir, MD Pathology, on investigative approach to immunology. Investigating the immune system can be accomplished with a variety of techniques, and sir has narrated everything. Here, sir has precisely cleared the concept of immunity with its types, different immunoglobulins, with appropriate examples. Sir has explained antigenicity and immunogenicity, that is manifestation of the disease, in short. And sir has explained antigen-antibody reactions, which are used in laboratory for diagnosis in vitro. Here, sir has explained precipitation reaction, populations, then agglutination reactions, and the different techniques used in immunology, that is radial immunodiffusion, then immunofluorescence, then ELISA techniques, that is linked enzyme-linked immunosorbent assay, then CLIA technique for measuring different hormones and cancer markers. Sir has again explained immunohistochemistry for detecting the histological samples of cancers. Sir has narrated this vast and complex material in so short duration of time. So everyone has to concentrate each and every word said in the lecture. So disseminating immunological developments, immunological investigations, it provides truly in-depth examination of the latest advances in molecular and cellular immunology. So applied immunology here focused on development, activation, and control of innate and adaptive immune cells. So lastly, I would have to say, this all comes under our Vedic Shamatva, that is Vedic Shama Sharirani and Vedi Akshama Sharirani, where Granthakara has said, Vedic Shamatvam Nama Vyadhi Bala Virodhitvam Vyadhi Utpadaka Pratibandhakatvam. Thank you, sir. And thank you all Rognidan Vikruti Vigyan PG Association members for inviting me. Thank you.
Thank you, ma'am. Now I would like to invite Dr. Kiran Dule madam for vote of thanks.
Hello, am I audible? Hello.
Yes, ma'am. Yes.
Yes. Very good afternoon, everyone. Myself, Dr. Kiran Pramod Dhure, MD Rognidan, and RVPJ lifetime member. Today, I feel immense pleasure to take this opportunity to propose vote of thanks on behalf of RVPGA Naidaniki webinar. Today's webinar, renowned person and speaker, respected Dr. Mayur Bhosle sir, MD pathologist, given a very informative and knowledgeable lecture on topic investigative approach to immunology. Respected sir, thank you so much for your valuable time on sharing with us your immune knowledge and clearing all concepts related to immunology and immunological methods. Thanks to respected Dr. Deepali Amble ma'am as today's webinar chairperson and suggesting some valuable information. Thanks to Dr. Mrunal Bhoyer ma'am for introduction of organization. And also thanks Dr. Geeta Sathavane ma'am for welcome speech. Thanks to Maharashtra Council of Indian Medicine, Mumbai.
Hello.
I think there is network issue.
Yeah.
Yes.
So rest I complete from my side. I thank everyone for participating in the webinar, and special thanks to Dr. Mayur Bhosle sir for sparing his valuable time with us. And I hope in future also he will cooperate us whenever whenever our audience will require the special topics. I think Dhure madam had joined again. Anyway, it was a very good lecture all in all, and with that covered in very short time. Thank you very much, sir, and thank you, Deepali madam, also. And thank everyone, and declare this meeting is over, and people can